And I'm happy to join TechSource and Asana Systems today to talk about MATLAB and Symbology in the context of modeling for drug discovery and development. So before I start, quick overview of the agenda. So what I'd like to show you is what Symbology can do to help you for your modeling efforts in the drug development process. And I'm going to illustrate this with two case studies, one about type 2 diabetes, and it's going to be a quantitative systems pharmacology case study. And the other one is going to be a case studies about optimizing the dosing amount of a molecule, of a large molecule here with TMDD, the target-mediated drug disposition model. And that is probably a bit more PK/PD type of application. Finally, I'm going to talk about how you can not only build your models, but also share them with others that are not necessarily modelers and not necessarily people who are also keen to program. So we're going to see how you can share those type of application and share simulation capabilities. And finally, we'll talk about the community, how we can support you, so the user community, but also ourselves, MathWorks and TechSource, obviously. And obviously, at the end, we're going to have some time for Q&A, hopefully. I hope so. I'll try to make it work. But also, in the meantime, if you do have any question, feel free to ask those questions and post them on the chat. I'm going to have a look from time to time on the chat to see if there is any urgent question, and if not, I'm going to answer them at the end during the Q&A. All right, so let's talk about Symbology and what it can do for you or to help you in your modeling efforts in drug development process. But before I do so, I wanted to introduce you a little bit to what it is good for, in which context. So this might be concepts that are already known by some of you. If that's not the case for everyone, I thought it might be good just to talk really in a minute about what pharmacokinetics and pharmacodynamics is, which is basically the basis or the basics of the modeling concepts that Symbology is implementing. So pharmacokinetics, or PK, is about knowing how a drug is going to be absorbed or metabolized by the body. So basically what the body is doing to the drug. So what you typically want to know is to have a time course of the plasma concentration of a given drug over time. So obviously how it's going to change in the dynamics of it. PD, the pharmacodynamics, is more about what the drug does to the body. So what you'd like to have is to have a concentration response curve of your drug that is going to give you how effective and starting from which concentration a drug will be effective. PK/PD is about marrying both concepts. So getting really a joint model of PK and PD that would allow you to have a time course or describe the dynamics of the response over time of the body after giving a specific drug, giving a bolus or any type of dosing schedule. Right, so PK/PD is really the center of what Symbology is good for. So there's different types of modeling here also in PK/PD that I'd like to address and show you where Symbology has a big role to play. So on the from left to right, we're going to increase the model complexity. And I wanted to separate PK and PD models because you'll see that there are different methods you can use for both of those applications. For the pharmacokinetics, it starts with small compartmental models, very standard models. You might know 1, 2, 3 compartments. Those are still differential equations in the background, right, that described the dynamics of the concentration of a drug in the plasma, for instance. But those are kind of gray box. You're not trying to describe all the mechanisms behind the absorption of a drug, for instance. But given or depending on the questions you'd like to answer, it might be useful to go more into the details of the mechanisms behind this absorption or behind the excretion of a drug. This is what models like the PB/PK models, physiologically based PK models, do. So here you're really trying to describe how a drug is going to be distributed in the body in the different organs. So those models, as you can see, they tend to be larger And here, this is not even a large PBPK models. I've seen models with just really thousands of state variables. So it can grow really, really large. It really depends on how the molecule is going to be absorbed in the body and where you think it's going to go. So here on from left to right, we're not only increasing the model complexity, but we're increasing also the model specificity to a given drug. From the PD part, there's also very simple models that are, again, not mechanistic. You're not trying to describe really the mechanisms of action of a drug, but it's still trying to describe it with an equation. And this is what, for instance, indirect response models do. They're fairly simple. It's kind of a gray box model too, but that works too. And this is very small and containable number of parameters as well. But you can also here go in much more details in the action of a drug. So basically, you would really detail how a disease or the processes involved in the disease look like, and have a very specific and detailed site of action of your drug. So this is typically what you might have heard of systems biology. What those systems biology models do. So mostly they're not trying about talking about drugs, but they are trying to find out the reaction networks specific to given pathways, like activation of a pathways after the reception of a growth factor, for instance, or an hormone, or whatever that would be. But really, you're trying to go into the details of actions of a protein with another one, or interactions between proteins and other compounds you can have in a cell. So here, again, you're going to into much more details. This is really specific to the disease you're looking at, and this can grow in complexity. So this is what quantitative systems pharmacology is trying to do as well. Typically, the PD part will be mechanistic. You're going into the details, and you would add some PK part also to go to the pharmacology world, and not only systems biology, where it's more about understanding the biology and not so much the disease or how to treat the disease. So here you see there's different complexities, different types of models, and you can really mix them. You could have a small compartmental model for the PK with a very mechanistic PD part. You could also have a PBPK model for the PK part, coupled with a simple PD or coupled with a mechanistic model. So really those models can be very, very large. So that means as well that you want to have a modeling framework, a software that allows you to build any kind of model, and that can grow also in complexity and still able to simulate your model even though it is growing in size and complexity. So where are those techniques used in the drug development process? It's more a reminder probably for most of you, but I'm going to separate that into really 3 phases only. The first one will be the discovery phase, where it's about identifying a drug target. Then come the preclinical studies, mostly done with data from animals, where it's about getting to see whether a drug could reach the target as expected, and try to find a safe and efficient dose for the first in-human studies. That is going to be in the clinic, one phase of the clinical studies that is obviously in humans. So here you're trying to assess really how the drug is going to be absorbed, metabolized, etc., by or distributed in the body. You're going to assess how safe this drug is and how efficient it is on humans. And finally, you're going to try to define the target population. Are there any specific subpopulations that might be really responsive to that drug? Are there any other that don't show any response, for instance? Are there any characteristics that might be lead to side effects or toxic effects? Right. So this is in that phase where you would want to try to figure out those kind of things. So where are those methods I showed previously used? Systems biology would be mostly used in the discovery phase, because here you're trying to find the drug target. So you really need to go into the details of the mechanisms involved or the processes involved in the disease you're looking at. And then for the preclinical studies and clinical studies, you would use mostly PK/PD or PBPK-PD, depending on the questions you like to answer. If it's about safety, for instance, or if it's about ADME, how the drug is going to be distributed into the body, you need to go into the details of these. So PBPK models might make sense. So it might look like those two types of models are uncoupled, and this is partly true until QSP showed up. And this is the idea of QSP, to try to bridge the gap between both methods. So try to use what has been done in systems biology, the whole work for the PD part, and we use that also in the later stages of the drug development process, combining it with PK model or PBPK model, for instance. So QSP has been largely used in preclinical studies or in the preclinical phase. I think there's also some efforts to try to see if it could be used also in later stages, like in the clinical studies. Okay, so once we know what that is and where they are used, so what is the typical workflow in those type of applications? And again, we're talking here about modeling and simulation. So the first step would be where you would get some data. You would need some data at some point. If that's data that needs some cleaning, you will need to clean it. And if that's a PK data, you might want to compute PK parameters using non-compartmental analysis. In terms of modeling, then you will need to specify first the model. That usually comes from what you know from the disease, from the literature, from the papers you might read. There might be also some previous models you might be able to reuse. But then you need also to adapt this model or calibrate it to your data, to the questions you like to answer. So that's where you will want to estimate some of the parameters where you're unsure about the values, or you know that you have to change the values, and get then a model that is really realistic for your use case that you could use for analysis. So of course, before using that model, what you'd like to do is to make sure that this model is realistic and get some confidence about that model. So that's where it might be useful to compute confidence intervals on the predictions and on the parameters. But once you know that your model, or you think your model, is realistic and can be used for further analysis, then you would apply it to what you have to do. That could be try to find out a good dose amount or dosing amount for your drug, evaluate the toxicity of your drug. Could be simulating virtual populations to try to figure out which population might be a good target. Could be target identification. So in the discovery phase, try to find where the drug or which process the drug should inhibit, for instance. So those kind of things. I think the applications are really broad, and this is really not an exhaustive tier of applications. Okay, just as a quick reminder, I had opened a poll here. I see some of you have answered it and many not. Maybe I can just restart it. I think it's still running now, if I'm not mistaken. So if you get a chance, please open the poll and answer those questions. That'd be useful for us to understand what your application would be. Okay, so what is Symbology in that context of drug development? So Symbology provides you apps and programmatic tools. And this is important because sometimes people think that's really just a user interface, but no, that's both. Provide apps and programmatic tools to build a model, and we'll see how it works. But you basically build your models by drawing in it, by drag and drop of different components. And this is really great to exchange and talk about the processes that you include in your model with experimentalists and biologists, people who might not be modelers, and so it's not really necessary for them to see the equations, but it's more visual and speak to them when they can see the diagram of the interactions involved. Symbology has been built primarily at the beginning, at least for systems biology models, so large models, and now it's focusing on QSP. But that means also that it's good at fast simulations of models. So there's a lot of work in the background to be efficient in simulation, because this is really the ground stone of everything you would want to do with that model. So that might be estimating the parameters. So Symbology also provides tools for that, or any other type of analysis you might want to do afterwards. That might be sensitivity analysis, for instance, like global sensitivity analysis or local simulating virtual patients or virtual population, for instance. To give you a quick idea of where this tool is being used, and then I'm going to show you that in case studies. I wanted to pick up a couple of papers from the literature from people we know of, people who have used Symbology in different types of application. And this one is about QSP, so systems pharmacology. This is a work by Zhu and Al from Jensen. What they wanted to do is have a look at factor XA. This is a good target for anticoagulation to prevent venous thromboembolism. So far, there's no model that can be used to do some prediction in the clinical context of the behavior of those inhibitors. So this is what Zhu and Al tried to do. They tried to develop a model based on the knowledge that they have from the drug discovery stage. And also they wanted to go further and then take that model and try to figure out, for instance, kind of parameters that might be important to determine the drug effect. So they've done this work, published it. So now there is a model available to make predictions. That's already a good thing. Plus, they could see that there's different kinds of parameters that are important, and not only everything that has to do with the drug, but also obviously the interactions that are embedded, and they go obviously more into the details of that in the paper. So if you're interested, I think you get the PDF of the presentation at the end, so there is a link also to that paper. Another one, QSP model, is from AbbVie, from Klausnitzer and Al. And here they were interested in understanding or in building a model of the Alzheimer's disease, because so far or before that paper, there were some mechanisms that were known separately, but there was no comprehensive model of the whole system. So they wanted to try to figure out if they could model everything that the current knowledge and build it into one single comprehensive model. So they've done this and published this work also as well in CPT pharmacometrics. So I guess I'll leave you the details of what the outcome would be for the biology, but the good thing is, again, you have a model with the comprehensive picture of what's going on, at least of the knowledge available, that you can use also for prediction because it has been calibrated. And almost the last one, but here it's a slightly different application. It's a paper from Roche, where it's here not about systems pharmacology, but it's about PBPK modeling. And the authors were actually interested in monoclonal antibody, which are largely used as a therapeutic entity. But there's still, or before that, there was still not much knowledge about the clearance or quantitative values really of the tissue clearances of immunoglobulin G antibodies. So that's what I wanted to do. Wanted to see if they could take some mouse data for labeled antibodies and see if they could determine those clearances. So they've done that. They could use parameter estimation using their data, build the model, and have it really specific to their work, and determine then the tissue clearances in the mouse in the unit of milliliter per day. So here we're really talking about a parameter you can reuse then in other models if you're going to simulate and work in a preclinical context using mouse data. Okay, and really the last one, and here it's not about research, it's about teaching here. I wanted to mention this because I said Symbology is both an app and programmatic tools. So that means that you can use the app to build a model, you can use an app to simulate, you can use an app for the parameter estimation, for instance. which really is a great advantage when it's about teaching. So here the students don't need to have any programming language, and they don't need to take care of how to declare a variable and to write the equations, but they can really focus on the model building, on choosing the kinetics, and then on running the simulations and trying to interpret the results, really. So here they can really focus on the modeling, and this is what Jagesh from Harvard Medical School liked, and this is why he's using also Symbology for quantitative pharmacology course. So there is a technical article also on the MathWorks website about this. So if you look for quantitative pharmacology, Harvard, even if you tap that on Google or any search engine, you would find it. But it's also linked here on that slide. Okay, so I've talked a lot about what Symbology is in more the theoretical way. Let's go now to the tool itself. And I'm going to show you a couple of examples. So the first one is going to be about type 2 diabetes. So let's say that here we are a pharmaceutical company. We're investing or investigating in new targets for type 2 diabetes. So we have one candidate. I would just like to make sure that there's no really meaningful reason to abandon or give up that candidate, and that we can go further and investigate it further. And I think this is an important concept also to see really early if it's going to work. So we are a modeler. We're given a small amount of time, so 4 weeks is not much to investigate if this target is feasible, if it makes sense at all. So we have no data because it's really pre-work, and we're going to have to make some assumptions. So one of the PK profile, and we're going to probably have to use also existing models because in those 4 weeks it might not be easy to build a new model from scratch. It's important here to understand again that this is more it's a very early stage of the drug development process, and it's more about failing early. So I've heard that a couple of times in talks. One I could remember was from Neil Benson. At the time, he was at Pfizer, and this is something he said multiple times, that it's important to fail early to see if a candidate is actually a bad candidate early, to avoid going to later stage of the drug development that are very, very expensive. It's a lot of time, money, and might be dangerous also if you're going to, you could even go to the clinical study without knowing that this is actually not working. So failing early is also important in drug development. So here we're not going to try to have a final model we would submit to the FDA, but it's more for us as an internal decision-making step. So we are lucky. There is a model we can reuse from the Kobody group about the processes of the glucose absorptions. So it's pretty mechanistic, it's pretty comprehensive, so we can base our work on that. We would need to make some assumptions. So first, that this model is adequate for the question you'd like to answer. And also, we know that it has a short-term prediction span, only on the order of days. But this might be still enough for us to see if the therapy would work. So this was just a model of the glucose insulin pathway only. So now, obviously, we need to include the SGLT2 therapy. And this has been incorporated by a consulting company called Rosa & Co., who work with Symbology and is specialized on QSP, on building QSP models, custom QSP models, using what they call the physio map that is based on the Symbology diagram concept. So here the assumptions are the SGLT2 effect are on the urinary glucose excretion. And we need to assume also the PK profile. Okay, so let's have a look at that model. So here I'm in Symbology, in the app of Symbology. And what you see here is a picture of the model. And when I say picture, actually it's pretty wrong. It's not only a picture, this is the model itself. So here, this is a graphical representation of the model, but we're going to see that this is the model itself. So this model has been well annotated, using colors, using some legends for you to understand what's going on. So if you always use from one model to the next one the same legend, the same color, so it's easy for you to understand what's happening in that model. Also, all the different processes have been separated, and you see here different numbers. That shows you the different processes or stages modeled in this model here. So the first one is how the glucose is being absorbed. So how the meal is being ground, is dissolved and then moved to the gut, or from the stomach to the gut. So what you see here is a state variable. This is something that will be described by a differential equation. That is going to describe the stomach glucose in a solid form. So the glucose in solid form in the stomach. Sorry. One thing I will do is just increase the size of this here for you to see that a little bit better. We have here in between a reaction. That means that here the solid form is going to be transformed into the treated form, and then it's going to be moved or transferred to the gut. Right. So all those here reactions are really the processes involved. Then we have another one that is about how the glucose is going to be utilized. So basically it's going to be redistributed also in the tissue, etc. There's another one about the insulin secretion as produced in the pancreas, it's going to be moved into the portal vein. Then it's going to go to the liver, so in here move to the plasma. There is typically a delay also between the insulin production and insulin action. This is what has been modeled in number 4. This is a small processes that includes simply a delay, and the delayed insulin is what's going to have an action. The last bit is going to be then the PK, so how the drug, how the SGLT2 inhibitor is going to be absorbed by the body. And this is a very, very simple model of one compartmental model, really. We're not trying to here have a detailed view of the mechanisms involved in that absorption, sorry. It's just a first order absorption. So this is what number 5 is actually doing. Once it's absorbed, once the SGLT2 inhibitor is in the plasma, it's going to have an impact on the urinary glucose excretion. What it's going to do basically is going to have an impact on the threshold of the glucose concentration, starting from which is going to be excreted. So this is the mechanism of action here, and we can only have those kind of description because we have a QSP model where we have some more detailed description of what's going on. Okay, to give you an idea, we see here glucose excretion. This is basically the reaction rate defined here for the glucose excretion. If we have a closer look at glucose excretion, here we'll see that it is first a little bit small, so I'm going to increase it, and it's here. It depends on the plasma glucose that must be above a Reynolds threshold. And then there's some parameters that define the glucose excretion. If we have a look at the Reynolds threshold, that is here and defined with that equation here. And I'm going to increase the size of it again. It's a pretty long equation, but you'll see that it depends on the plasma SGLT2 inhibitor. And that's basically really the only place in that model where the inhibitor is going to have an impact. So here, the more we have, the lower the threshold is going to be. Right, so that's basically the mechanism of action of that model. So I said this is not just a picture of that model. This is really the model itself. And this is true now. We can have a look at the equations. those are the differential equations generated automatically by Symbology from that diagram. That means you don't need to write the equations yourself. You can if you want, that's fine. But you can just think in terms of processes and the types of kinetics you want to have in those reactions. And then Symbology will take care of the generation of equations for you. So basically you would, to build that model, you would drag and drop component from that library here. Species are state variables, so that's the same as the blue one here, just that we change the color. And you can include a reaction in between. That means that species 1 is being transferred into species 2. And here I just defined a parameter, let's say k, to define the kinetics, so how fast this transformation is going to occur. And that's basically it. If I go back to the equations now, you'll see that there's other equations that will show up in a minute, and here they are, those two. Those are the differential equations that were generated automatically by Symbology from the diagram. I think you understand now. It's really more about having a good view of the processes involved in your model, and Symbology will generate the equations for you. I'm going to remove this because this otherwise will give me an error for what I like to do. And another thing that you can also do, for instance, in Symbology is define units, and we'll take care of the unit conversion. Okay, so that was for the model. Now we said, okay, we are a modeler and we like to make sure that the SGLT2 inhibitor is actually a good candidate and that it leads to a reduction of the glucose concentration in the plasma. So what we'll do first is simulate healthy patients. They have a profile of what the glucose concentration would be in the plasma. And for this, we're using some variants. Variants are, let me go back to this quickly. Variants are a collection of parameter values to describe a specific condition. So here we have one for healthy patients. So we have to give those parameter values, but we can save them in a variant and then use one variant or the other one. So here I'm choosing the variant for healthy persons, and I'm going to give also this person 3 meals, right? As you would expect, defined as doses in the model. If I do so, we see the profile of the plasma glucose. That's the one in the middle in milligram. The one at the top is the PK, so how the drug, the SGLT2 inhibitor plasma concentration looks like. So here we're talking about healthy patients, so we don't give any dose, we don't give any therapy. So that's fine, everything is 0. And here you see how the AUC of the plasma concentrations over 1 day looks like. So this is for healthy patients. Now let's simulate a type 2 diabetic patient. And I forgot one quick thing. I'm sorry about that. Let me resimulate the healthy patient first. And I forgot to overlay the results. If there is here on the right side the possibility to overlay results, that means if I rerun with some new conditions, it will keep the old plot and show me both plots so that I can compare both conditions. Okay, so you see the one with the dashed line, that's now the type 2 diabetic patient. We see that at least here at the bottom and also on the top that the plasma glucose concentration is much higher, especially the AUC. You see really the large differences over 24 hours. So what we like to do is actually bring back this curve here to that level, or see at least if this inhibitor is able to do so. So what I'll do is now include the dose for my inhibitor and rerun, and we'll see what happens. Okay, so that's the one with the dotted line. So you see there's not much difference. Now that didn't lead to a big reduction of the plasma concentration at the AUC and plasma concentration over time. What would it be? So you might say, this is fine, I'm confident my model is okay, so it's just maybe because my candidate is not a good one and I should reject it and stop investigating. So it would save us all a lot of money if you think that this is the right answer. But maybe we need to go more into the details of that model and see if all those parameter values are actually fine. And we did so, and we realized that the one specific parameter that is called GFR, that is the glomerular, I always forget that name, sorry, glomerular filtration rate, basically the flux of the flux that goes going through the kidney. So basically how fast the glucose would be excreted in the urine, that this has been underestimated, and not by a small amount, but we are thinking about tenfold. What I can do is select that parameter here. I can drag and drop that parameter here in the Explorer, and what I get is a slider. So I could just move the slider and see what happens if I change that value. I could also just double click here on the current value of that parameter. I'll remove a 0 to multiply by 10, press enter, and it's going to resimulate for that given value. So here, that's the one you see, the dashed dotted line. This is the new one with the new GFR value. So for that new GFR value, we have some literature references, right? It's not just coming out of the blue, but we had to look for it and see if there's some more references we could find, because we had a suspicion that this parameter was not right, not calibrated correctly for our need. As you can see here, at least the inhibitor is not, it does not provide the same level as previously as for healthy patients, but it does decrease substantially than the plasma concentration. So it looks like it's a good candidate for us. At least we don't see any reason to reject that candidate, and we would go further into the investigation. Okay. All right. So here that was pretty easy because we had some parameter values already in the literature. So we had to build the model and run the simulations and run those what if scenarios. What happens for a type 2 diabetic person? What happens if I give dose of the HLT2 inhibitor? But in some other cases, you might need some other techniques. You might need to do some other things. Like here, we're going to talk about another case studies that requires more methods. And here we're trying to find dosing amounts of a large molecule using a TMGD model, so very small model. And that model looks like that. It's really just the drug binding the receptor or the target and building a complex. And that's basically it. So it's eliminated here on all state variables. So the target is eliminated, the complex is eliminated, the drug. The receptor is being synthesized as well. And the drug here will receive some dosing we need to define. There is also a variable called target occupancy. That is the percentage of the target that has been bound to the drug. And this is what we're looking at. This is our observable, so to say. So what we like to do is find a daily dosing amount. So we know we want to give only one dose a day, so that this target occupancy remains between two thresholds. 15%, that is the efficacy threshold. So below that is not efficient. And it has to be below 85%. Otherwise, it's we have safety issues. Right. So we're trying to find out this amount. So we will simulate different dosing amount and see what works and what remains between those two thresholds. So for that, let me close the previous model. And I'll open the new one. And you see here, I'm selecting a file that is called SBproj for Symbology project that always contain all this. Contains my model. It contains some data that I might have, and programs that are things that I do with that model, like simulations and parameter estimation, etc. So each time I reopen that project, everything is there. So I can also share the project with collaborators, and they will see the same thing as me. So again, model is pretty simple. That's the as before, it's the same visual view. And here the difference is that I have some data. So here I can choose my data set and drag and drop, and I have some data that shows me some measurements of the drug, of the target, and of the complex in the plasma for given time points for a number of subjects or mice. And here we have 12 of them in total. And we can also have a look at this data. Apologies, I'll change the view here. So we're going to have the plot of this data. That's the dots here. And currently, the lines that you see are the current simulations given the current values of my model. So if you see, it's completely off. It's not reproducing my experimental data, so it's not a model I could use for predictions. But one thing I could do is obviously take some of the parameters here, KL, KON, create some slides, and try to figure out manually and get those lines closer to my data point. But this is manual work. Might be good for just one parameter, but if you have more, that's really difficult. So instead, what we would do is run a parameter estimation in optimization of the parameters to fit my experimental data. And this is one of the programs available in Symbology. So in the previous case study, we used simulations, but now we're going to use fit data. And there's many more. There's non-compartmental analysis you could do. There's a scan by scanning, sampling parameter values, and running the different simulations, running to steady states. There's a lot of things you can do here. But we'll focus on the fit data task or program. And I have one that is prepared here, where I tell my model here in my column drug M and M, that is the measurements that I have. This corresponds to the species plasma.drug in my model. Plasma.receptor is what's called target and name in my data set. So I need to map basically my data to my model. Then I need to list the parameters I'd like to estimate. And there's different methods I can choose, but probably don't need to go into the detail of that. What I'll do is simply click run and show you how it looks like. So now this is going to be prepared. My model is going to be prepared for that, so the whole algorithm. And we'll have a look at the log likelihood. This is what we want to maximize. So basically, this is a quantity that is the inverse of the differences between my simulations and experimental data. Mostly with a minus, more than the inverse. So this is done already. So we saw how the different parameter values are changing during this optimization problem or using this fit, right? Until getting to a value, and that is the estimated value. So I'm going to close that one and have a look at different plots that we have. So here, this is how it looks like for drug receptor and complex on each row. You see the dots, that's my experimental data, and the black line is my simulation, and the blue that you see around it, might be a little bit small to see, is what's called the confidence intervals for the predictions. So it really gives you a measure of how safe or how the predictions look like. Okay, so we can have a look also. Now this is the previous plot, sorry, and here this is the final also comparison between experimental data set and simulations. And once I have that, what I can do is go to my results here. That's the results of this fit we've just done. And I can create a variant from my estimated parameter values. And if you remember in the previous case studies, we used variants to simulate type 2 diabetes patients, for instance, or healthy patients. Now I could save that into estimated values and use that variance now for the rest of the analysis. That means that I have my model calibrated. So I don't need to do that because I've done that previously. what I can do now is run a group simulation, or sorry, not a group simulation, but optimized dosing, that is going to test different dosing amounts. This is what I do here. I take daily dose amount and I'm going to sample different values of this dosing amount between 0 and 300 nanogram. And I'm going to pick 31 values in between, linearly spaced, uniformly distributed into those three, or into those binaries, sorry. So if I click run, now it's going to simulate my model 31 times with different daily dose amount. So we leave it run. And I'll show you that in a minute. I have at the bottom of this task, or this simulation program, I have created an observable that is going to return me 1 if my target occupancy is remaining between those two thresholds that I've just defined, the safety threshold and efficacy threshold, and 0 otherwise. So after each simulation, it's going to compute that quantity and return me 0 or 1. And the result of that will be shown on this plot here in a minute. There you go. That gives me a plot of this quantity, 0 or 1, for all daily dose amount that I've tested. So now I see to be 1, I need my daily dose amount to be here around probably 80 up to 200. So that's my dosing amount window that I can use. That seems fine, at least for my target occupancy to remain between the two thresholds. So maybe to be safe, I'll pick something more in the middle here, maybe between 100 and 150. But that gives you an idea. Obviously, you can have a look at the simulations also that were produced. That's the 31 simulations. And here we're looking at the target occupancy. And you can see that not all of them remain between those two thresholds all the time, but some do. Right? This is what we see in this other plot here. Some do, and they are here. So here we've used the parameter estimation. We've used the scan program to scan over a given parameter that could be also parameters of the model. Okay, in the interest of time, I'll keep going. So what we saw here is first that building a model in Symbology is something visual. You can also do it programmatically. Right? It's not that you have to use the diagram, but it's a very good way to share also the and visualize the processes involved in your model to work with biologists or experimentalists. So while you would do that in a paper anyway to write the differential equations, here you can do it directly in Symbology, and Symbology will write the ODEs for you. Things you can also do, non-compartmental analysis, don't necessarily want to go into the details of that, but this is also included in Symbology. Other things would be, okay, we've seen that the parameter estimation to get the confidence intervals also of the predictions, but also of the parameters. There's different techniques that are included, like profile likelihood, for instance. Other things that are there would be the global and local sensitivity analysis. So local might be useful in the discovery stage to find their targets. And the global one is good to find out to which parameter a given state or given concentration would be sensitive. And that helps you to find out which parameter you should carefully estimate during the parameter estimation and which parameters you can just fix because they don't have a big influence anyway on what you measure in your model. So there's both really. So the local sensitivity analysis has been around for a while, and there is the global sensitivity analysis using Sobol indices or a multiparametric global sensitivity analysis. We had last week a webinar on the global sensitivity analysis, and it's going to be online very soon. So if you're interested, you'll soon be able to see that on our website. Another thing I'd like to mention again is Symbology is an app, but you can also work programmatically if you prefer, and that's typically what I like to do. You can still build your model in the app and then import your model in MATLAB to work with it using Symbology functions. So typically here, if you're looking at a simulation program of Symbology in the app, there is the equivalent also programmatically. So you can import your model first using SBio load project from the model that you've built visually. You can choose a dose. You can then set the simulation time to 3 days, for instance. And while you will click on run on the app, there is the equivalent SBio simulate to simulate that model given those conditions. And you can plot the results with SBio plot. So you see, really, there's an equivalence between those two here that's doing the same thing. But the big advantage of working programmatically is that you can really build your own custom analysis that might not be available in the app or to automate tasks. So quickly to show you who is doing that. There's, for instance, BMS, or Bristol Myers Squibb, who wrote a QSP Toolbox that is really streamlined for what they do, their QSP workflow, and they've published a paper around that. And they provide also the QSP Toolbox freely, so you can download it also from the MathWorks website, from the File Exchange. I believe it's also on GitHub. Similarly, there is also QSP IO, so for immuno-oncology. This has been published really recently in July by the Johns Hopkins University. And this is here a way to create a model that is modular programmatically. This is a Symbology model that is built programmatically. And then you can choose the models depending on the questions you like to answer. So this is a great way to build a really research question-specific model. And they've illustrated this with 2 examples of immune checkpoint therapy. Okay, and quickly now, model sharing. So if you're a modeler, that's great. You can work with MATLAB and Symbology, build your models. But in some cases, we've seen that there is a modeling group, people doing the modeling work, and the people who might use that model might not necessarily be the same people. They might not be modelers and might not know also about programming and MATLAB. So this, what you'd like to do is provide them still the capability to simulate, because you don't want them to go back to you each time they want to simulate maybe a new experimental condition or new dosing amount. So you can do that in MATLAB. You can build your user interface, and this is what Genentech has done together with our consulting as well. They've built a tool called GPKPD Sim that is based on Symbology, but it's just one single window that is streamlined to their process. And they wanted to have people who are not necessarily modelers that do their own simulation and run NCA, for instance, etc. So this is also published, and this is also available freely on the MathWorks website, on the File Exchange. Same thing for GQSP Sim, it's the same ID, also from Genentech, but here is also supposed to streamline their QSP workflow. So a bit similar to what BMS has done, but here, while BMS provided only scripts and functions to do that, here Genentech focused on a UI, on a user interface. And the last bit, and probably not the least interesting, is that you can also build your own applications. You can use App Designer in MATLAB to create a user interface, and this is included in the MATLAB-based product. Then you can compile it with the MATLAB compiler, put it on what's called the MATLAB Web App Server. And on the web server, you can upload it. There is a button, Upload App, that allows you to upload the application, and then the end user, could be anyone, just need to open the browser, Chrome, give the URL that you send probably per email, and start the application. There's no need to install anything. Everything is running on the server. It's great for you also to manage who's accessing what and updating also versions of your app, because everything is centralized on this web app server. So this might be very useful. If you're interested, please have a look. And I don't think I need to demo this because it's a bit late already. Right, so the really last slide. There is a Symbology community website, so you can feel free to go there. There's links to tutorials, video tutorials. You can ask questions that might be answered by other users, by us as well, by our developers also monitor this website. And you'll find also links to those QSP Toolbox, those GQSP Sim, and all those community toolboxes and apps that have been produced. So we link them on the website as well.